Good morning, happy Monday. Glad that you all made it here through the rain. Today okay, since you're wondering about this, the exams will be distributed on Wednesday. Not today. We're going to continue our discussion of sort of the basics of biological structures and some applications in nanotechnology. So last class where we talked about new material, which is now more than a week ago, we talked about protein structures and polypeptides, and then the use of sort of the unconventional uses of protein structures, for example, in flexible, stretchable, and biointegratable electronics. And today we are going to talk about nucleic acids, so DNAs and RNAs, and then nanotechnologies that use these, and actually a number of significant connections to not just the San Diego area, but also to UCSD in particular. And to start with, DNA and RNA, of course, is the information coding molecules of biological systems. And people that have been associated with UCSD have had a pretty important history in the history of the origins of biochemistry. And has anybody heard of the Miller-Urey Experiment? The Miller-Urey Experiment was an experiment performed at University of Chicago right when Harold Urey was transitioning from University of Chicago to UCSD, where the idea was, can you prove that biomolecules could arise from abiotic circumstances? So what Stanley Miller and Harold Urey, who is his mentor, did was combine ammonia, methane, hydrogen, water, hydrogen sulfide into a vacuum flask, and then applied a voltage, and this arc, the energy produced in this arc was enough energy to produce amino acids. And several variations of this procedure have been performed after this, and have even produced some of the DNA nucleosides as well. Not this exact experiment, but other similar ones. And this is Stanley Miller, who was a professor at UCSD for several decades, and then his mentor Urey Hall was not his mentor. Harold Urey was his mentor, and Urey Hall at UCSD, which is the first research building on campus, which is part of the chemistry department now. The problem with this, just in case you're wondering, this experiment was first done in the 50s and 60s. The problem with this is that these are reduced species, and in the pre-biotic Earth, it's probably not too likely that the atmosphere was reduced. It was probably an oxidized environment. So the current theory is that the prebiotic Earth had about 10 atmospheres worth of carbon dioxide in it, a bit more like the atmosphere of Venus is now. So really, we didn't start out with reduced species, probably started out with oxidized species that may have been converted through some kind of proto-metabolism using iron and nickel and manganese sulfide nanoparticles that are ejected from deep ocean smokers that have catalytic properties of taking CO2 and reducing it to biomolecules. So this is probably more like how the first biomolecules arose on Earth, but in any case, this was the first experiment, although the starting materials were possibly the wrong ones. Okay, these are the purine and pyrimidine nucleosides. This is A, G, C, T, and U, and you're probably familiar with this from Build 1 or equivalent. So you know which ones are purines and which ones are pyrimidines. Do not worry about the structures. I'm going to show a lot of structures in this in the first few minutes, but don't worry about them for the purposes of the exam. I just want to show you what A, G, C, T, U actually mean. You know which ones are purines and which ones are pyrimidines because purine is the shorter name, and of course it's the larger molecule. And pyrimidine is the longer name and the shorter molecule. So it's a reverse mnemonic, right? So purine must be the larger, or must be the shorter one. And that's wrong. So as long as you know it's a reverse mnemonic, you know which ones are which. So the difference here between DNA and RNA, the sugar is different. We'll show that in a second. It's deoxyribose in DNA and ribose in RNA. The other difference is that thymine is the base in DNA and uracil is in RNA. So all the T's are used, but they're basically the same structure except for this methyl CH3 group on T, but not on U. Okay, when you take the purine and pyrimidine nucleoside bases and you bond them to the sugar, this is the case of deoxyribose. So this is deoxyribose, and what's deoxy about it? It's deoxy because there's no OH group here. That actually makes it more stable against degradation in the biological milieu. Then you also have this phosphate group, and what happens is that these bases polymerize by a condensation reaction. So this phosphate group couples to the OH group here, which couples to the phosphate group here, which couples to the OH group here, and so on. And that's how the links in the chain forms. These are the RNA nucleotides. So the difference here is that we have a hydroxyl group on this carbon atom, whereas deoxyribose has no oxygen, or has no OH group. This is the linkage to which I referred. It's called a phosphodiester bond. So phosphodiester, sugar, and then the nucleoside is bonded here. So ATG, UGC, and the way that we describe the orientation of these chains is by this nomenclature of 5' to 3', which refers to the numbering system of the sugar. So so this is, for example, the 5' CH carbon atom. This is the 3'. So this whole chain, it goes from 5' to 3', and that's how we know. That's how we can arrange. That's how we know what orientation of a DNA molecule we're talking about. Why is this prime? What is the unprime? The unprime numbering refers to the numbering in the nucleoside base, so the purine or pyrimidine. That's why the sugar is called. But we never really use those numbers. We only ever refer to the primes. All right. So we know that DNA has a self-complementary strand. One is 5' to 3', and the other one is 3' to 5'. And when you match them up together, they form these DNA base pairs where the nucleosides bond by hydrogen bonding to a complementary nucleoside, and C goes to G, and A bonds with T. So if we have one, we automatically know what the other one is, and that's the secret of DNA replication, because you can split this down the middle, and you have this really complicated enzyme complex that synthesizes 2 new DNA strands by untwisting and repolymerizing the complementary strand from the template strands. This is a schematic diagram of the structures that shows you the dimensions of the hydrogen bonding arrangements that you have in AT or AU in the case of self-complementary RNA strands, and and GC here. What do we notice is the biggest difference between AT and CG? The number of hydrogen bonds. So AT only has 2 hydrogen bonds, and this is the hydrogen bond goes from a highly electronegative atom attached to a hydrogen atom, which bonds with the lone pairs on another atom. Okay, so AT actually is a weaker interaction than CG, and this is actually shown in calorimetry traces where the melting temperature of CG is actually, of all CG-based pairs, is actually higher. It takes longer to, it takes more energy to dissociate the chains than it does for AT because of the difference in the number of hydrogen bonds. Here are a couple of ways of showing DNA in schematic diagrams. So this is kind of the cartoon here, which shows the dimensions. The pitch of the DNA is about 36 angstroms, or 3.6 nanometers. The distance between two nucleotides is 3.4 angstroms. And then if you take a double helix and you twist it up, you get a minor groove when projecting this on a screen. So this is the smaller groove, and then you get a major groove. So minor groove, major groove. One misconception, and this is because this is the way that the DNA is always drawn, is that we tend to think of the AT and CG base pairs as being arranged with their axes parallel, the flat molecules with their axes parallel to the helical axis, but that's not actually the way that they are. They're more like a ladder. So the flat molecules lie flat in a plane perpendicular to the helical axis, like this. So the A's and G's and I keep doing that. The A's and T's and G's and C's line up like a staircase. So this is like a staircase. And then this is sort of the van der Waals-ish space-filling model. Okay, so DNA is self-complementary. DNA is complementary to another strand, and RNA, because it's superficially similar to DNA, actually has many of the same properties of complementarity. So in this case, this is a tRNA. So remember the movie that we showed about a week ago, where we showed the ribosome in action. It's like the most successful example of a nano machine, because it has actually assembled human beings, right? It's actually assembled biological structures using just quite simple building blocks. And those little things that went into the ribosome, those little things that were being grabbed by the ribosome that kind of looked like this, as in the assembly line, the little guys that it was grabbing are called tRNAs, and they're an RNA molecule that has kind of this that are self-assembled by these self-complementary strands. So you have A pairs with A and U pairs and C and G pairs that produce this sort of self-assembled structure. And down at the bottom you have an anticodon, which binds to the messenger RNA in the ribosome. And up here somewhere you have an amino acid residue that the ribosome positions in such a way to polymerize into the molecule. So the point is here that RNA has functional capabilities. It can actually form these secondary structures, much like proteins can, by self-complementarity. And here are just a few views of it. This is a schematic diagram that shows in a very simplified way the way in which DNA is replicated. In fact, it's It's so simplified that I don't want to leave it on the screen for that long, because one of the strands is polymerized quite easily, and the other strand actually has to be done piecemeal because you're asking the machinery to synthesize DNA backwards. Anyway, not important for us, unless you do a lot of molecular biology, in which case it would be. Okay, DNA, the point of DNA is to provide the blueprint, or the recipe. A recipe is probably better than blueprint because timing is important, whereas a blueprint is just, here's a human. DNA doesn't really work like that. It doesn't tell you, here's a human. It's more like, here's a human combined with all these time steps. So here is an example of DNA. DNA contains a gene, which is one protein coding region. From the gene is transcribed into a messenger RNA, and the messenger RNA finds the ribosome, and each set of 3 DNA or RNA nucleotides is translated by the ribosome into a different amino acid residue. So every three units on the messenger RNA corresponds to an amino acid. Then the ribosome produces this unfolded hexokinase chain, and then this is a peptide. So transcription, then translation. Transcription from one language to another. Transcription from one similar language to another, that is DNA to RNA, and translation from 2 totally different languages, RNA to an amino acid sequence. And then this is not the native structure, of course. You get a secondary structure. Sometimes this involves help from other enzymes that help to fold this structure into its final state. And the genes can be mutated sort of at will using genetic engineering. Okay. One of the most important inventions of the last 40 years was certainly polymerase chain reaction. A polymerase chain reaction is how you start with DNA and you end up making many, many copies of it, or they call it amplifying the signal. And the key to this process was the discovery of a temperature stable temperature stable enzyme that can polymerize, that remains stable even at elevated temperatures. So the idea is to mix together 4 components: the target DNA that you want to copy, the primers, which are complementary DNA fragments that bind to the beginning and the end of the sequence that you want to polymerize. Then you add the nucleotide. This is the starting materials. So deoxy ATP, deoxy CTP, deoxy GTP, deoxy TTP, from which you can polymerize a DNA chain. And then you add this thermostable DNA polymerase. And the thermostable DNA polymerase is derived from a deep ocean bacterium that survives in very hot environments. Then you buy a machine, a PCR machine, that's claim to fame is that it can increase the temperature and then decrease the temperature and do this many, many times. That's all it has to do, and you'll see why in the schematic diagram in the second slide. And basically, you melt the DNA. That means it dissociates into 2 chains. Then you cool the separated strands, and at that point, the primers that you've added anneal to the target DNA and also prevent that DNA from reassociating with itself. Then the polymerase extends the primers in the 5' to 3' direction. Then after a round of elongation is done, you repeat the steps. And here's, oh yeah, this is the fun part. This was invented by Kary Mullis, who won the Nobel Prize in 1993. And he was quite an eccentric scientist who, among other things, claimed to have been visited by a fluorescent green raccoon, and although he is a well-known user of psychedelics, he insists that he was not tripping when he saw the fluorescent green raccoon. Anyway, okay, so this is the. They say that that biotechnology can be divided into two eras: the before PCR era and the after PCR era. You really can't do much of anything in molecular biology without being able to copy DNA of interest. So here it is again. You have the 3' end and the 5' end, and then you have, say, a gene in here. And you separately synthesize some short chains that are complementary to the ends here. And these short chains are called primers, and DNA is always synthesized in the 5' to 3' direction. So then what you do is you have this thermostable Taq polymerase, which is the thermostable from the deep ocean bacteria polymerase that extends the chains. Okay. But now you've extended the chains, you still have primers in the mixture, and you just keep repeating the steps, and then the primers anneal to the old strand plus the copy and the old strand. Then the polymerase extends the chains again and again and again and again. After 20 cycles, the target sequence can be amplified about 1,000,000-fold. It's a little bit like step growth polymerization in that sense, or having a penny and doubling it every day. In a month, you're a millionaire. Then you have enough stuff that you can actually work with and do chemistry and use these sequences to add into other genes to do genetic engineering and so on. One of the things that you can do is you can do DNA fingerprinting. So DNA fingerprinting is a technique that is used extensively in the criminal justice system and in Law and Order and CSI and Law and Order SVU and Law and Order Regular and Law and Order Criminal Intent and Law and Order Los Angeles, which I think only lasted for 1 year. Okay, so what DNA fingerprinting does, and this is not whole genome sequencing. So there are a few different levels of sophistication of using someone's DNA to get analytical data. And we'll talk about all three of them. There's DNA fingerprinting, which is by today's standards not really that sophisticated. It doesn't really give you that much information. Then there's SNP genotyping, which is single nucleotide polymorphism genotyping, which tells you in the protein coding regions what differentiates you from everyone else in the Homo sapiens species. And then there's full genotyping, which gives you not just the protein coding regions, but all 3 billion, 3 times 10 to the 9 base pairs in your entire DNA, only a small fraction of which is actually protein coding. Okay, so we'll talk about that in sequence. So DNA fingerprinting is based on the idea of short tandem repeats that exist that are not useful necessarily, although I don't know, it's probably too early in the history of science to say what's useful and what's not. But we have these short tandem repeats in several different locations in our genome, and they can be cut out by something called restriction endonucleases. And those short tandem repeats from somebody's genome have different lengths. And every human being has different combinations of different lengths of these short tandem repeat sections. So what you do is you run them on a gel, and this separates on the basis of charge and size, and then you stain the gel, and you get a pattern that looks like this. And this is basically the sizes of the short tandem repeat sequences in your genome that are different from other people's. It doesn't tell you if you have blue eyes or brown hair or that you can or can't taste cilantro, or is it that you think it tastes disgusting, I guess. How many people think cilantro is disgusting? Okay, that seems about right. Okay, so it doesn't give you any of that information. It's just purely how long are these short tandem repeat sections in your DNA. And then you match one to the other, and it tells you if if that's so-and-so's blood at the crime scene or hair at the crime scene or whatever. Apparently, there was a famous case where a guy who knew something about phlebotomy was convicted of a crime, and he had access to other people's blood, and somehow he was able to implant a tube of someone else's blood in a plastic sheath in his own forearm. And then when they took the blood to do the DNA fingerprinting, they pierced the dummy blood and then did the PCR on that and did the DNA fingerprinting assay on someone else's blood, and he was exonerated. But then he was no, not exonerated. That's if you actually didn't do it. He was released, but then somehow he told somebody because he was so proud of what he had done, and then he was rearrested. All right, another cool example of biotechnology, especially as it relates to work done at UCSD, and much of this work was done at UCSD, are the use of using genetic engineering techniques to take a fluorescent protein and then put it into the genome of something that shouldn't be expressing that fluorescent protein. And many of these, the most famous example is green fluorescent protein, GFP, which undergoes a rearrangement in some of its amino acid residues, about 3 of them, that give you a conjugated unit that give you fluorescence, just like a dye or a quantum dot. I mean, it absorbs light, goes up, thermalizes, emits a fluorescent photon. So anyway, there's no special chromophore. It's just a reaction within the amino acid sequence itself that is susceptible to forming this chromophore. So anyway, the green fluorescent protein gene can be added to pretty much anything you want, and you can do things like you can target the locations of genes. You can target the locations of proteins within the cell or within the body, and this won the Nobel Prize a few years ago. But here's a gallery of kind of cool images. So the fluorescent proteins have this beta barrel structure, and then in the interior to the structure is this isn't actually a chromophore that has been captured from the environment. This chromophore, which is what the space-filling model is actually derived from a self-reaction of a few of the amino acid residues. And don't worry about the mechanism. Honestly, I had to look it up yesterday, and I forgot it already. So, you get a dye molecule just based on the way that the amino acids react with each other. And you can have different fluorescent proteins that have different conjugation lengths and protonation states that give you basically all the colors of the rainbow. So you can really localize one protein in a biological environment and differentiate it from another protein in a biological environment. So here are bacteria. This is a C. elegans worm that has this purple fluorescent protein and green fluorescent protein in different cellular structures and locations in the body. You can also do really useful things, like make GloFish. Fascinating. I think you can buy these. Oh yeah, okay, and here are the scientists who discovered the fluorescent proteins. Roger Chen at UCSD, who passed away a couple months ago. Osamu Shimomura at Boston University, my undergraduate alma mater, which makes me special because he invented GFP, and Martin Chalfie at Columbia University. Okay. Oh yes, fluorescent guppies, or whatever they are. Zebrafish, probably. And gigantic glow-in-the-dark rats. Just kidding, I don't know that they're gigantic. But I'm pretty sure that they shoot laser beams out of their eyes and can burn buildings to the ground and stomp on taxi cabs and stuff. All right, Human Genome Project also has a very close connection to San Diego. So the Human Genome Project had two fronts. One was the public effort, which was led by Charles DeLisi, who was the former director of the Department of Energy, Health and Environmental Research Program, and the Dean Emeritus of the Boston University College of Engineering. So basically everything cool was done at my undergraduate alma mater and your undergraduate alma mater. And this is Francis Collins, who is the NIH director. Actually had the opportunity to see him speak in December, which is pretty cool. And J. Craig Venter, who is a local businessman who ran Celera Genomics. Actually, Celera Genomics was the private company that published the first full genome, and it happened to have been of the founder, Craig Venter. So Craig Venter first goes as the distinction of being the first person to have his DNA entirely sequenced. He's also now the co-founder of Human Longevity, which is a company that uses a genomics approach to eliminate age-related diseases. What are we going to do after that? I don't know. And he got his PhD at UCSD. And just to show you, this is the J. Craig Venter Institute. We are, where are we? Somewhere here. Close. Okay, now for the longest time, it took a very long time when the Human Genome Project was started. It took a very long time to take DNA and to determine what all the 3 billion base pairs were in order. So until the technique of next generation DNA sequencing was developed, and this is most notably developed by a local company called Illumina that you've all probably heard of, and it is also very close to here. And the idea is that you bust up the DNA of interest into multiple, well, first you do PCR on it, then you break up the DNA into multiple overlapping fragments. By multiple, I mean hundreds or thousands, more than thousands. I don't know how many they do right now, but it's a lot. So you take the whole genome, you bust it up into many overlapping fragments, and then you immobilize them on a surface, on a micropatterned surface. Then you flow through fluorescently tagged DNA bases that are fluorescently tagged, and you know which sequence was in which spot on the slide or the wafer that you're doing this experiment on. You flow through the bases, and then when the bases bind to their complementary strand, they light up. And if you're doing this over a large enough sample size, and you keep flowing through new bases, and then you take a photograph of each time all of the spots light up because a new A, C, G, or T has bound to the immobilized DNA fragment, then you know that that was the next letter in the sequence. And if you do this enough, and because they're overlapping little fragments, you take all the overlapping fragments, you find the regions of similarity, and then using a computer, you can stitch together the entire genome. So this is the schematic diagram of what I rather ineloquently tried to explain to you. You take the DNA fragment of interest, then you flow through fluorescently labeled DNA nucleosides, then they bind. Then you do some chemistry, and then you do it again, and then the A binds to the T, then you do some chemistry, and you flow through the next one, then the C binds to the G, and so on. And each time you get a binding event, this thing lights up with that color. So if you take these red, blue, yellow, and green spots that show up, and this is a small fragment of a much larger wafer that has many of these broken apart DNA fragments that are partially overlapping. And you do image analysis, and you can read out the sequence based on each frame. Does that make sense? So then what you do is you take, each one of these spots here corresponds to a different fragment. Then if you have the letters and some of them overlap, you already know they overlap because you broke up the DNA in such a way that they would overlap. Then you can piece together the entire genome. This is called shotgun DNA sequencing, and variations on this method are the basis for really high throughput DNA sequencing. Incidentally, Illumina is also a mile from where we're standing, so a lot of San Diego and UCSD very close connections to genomics. Here's Illumina here. Okay, now to get a lot of good information about what makes your genome different from someone else's genome, you don't need to get all 3 billion base pairs. What instead you can get is called the SNP genotype. That is the single nucleotide polymorphism genotype. And the SNP genotype represents the areas in the genome that make you different, at least within the protein coding region of the genome. Okay, this is not the same thing as DNA fingerprinting. It's quite a bit more sophisticated than DNA fingerprinting because it actually involves getting information about the nucleotides in your genome. And what it's useful for is building phylogenetic trees between humans and all life on Earth, identifying genes associated with disease, developing therapeutics, personalized medicine, tracking the path of human migration, so full genotyping, not to be confused with the SNP genotype, which is only 62 million SNPs as opposed to 3 billion full base pairs. So humans on average share about 99.9% of DNA, and what's left over is the SNP genotype. Okay, so here is a pie chart showing the distribution of DNA. And what you'll notice here is that the protein coding genes are only a very small fraction, less than 2% of the total. Introns are, it's a little bit misleading to put introns as separate from protein coding genes because introns are actually segments of non-protein coding genes that exist, of non-protein coding segments of the genome that exist within the protein coding genes so that the protein coding genes can be spliced around and used to encode many different proteins just from one gene. The introns give you the space to move the DNA around. DNA, it's important to know, is not just an information carrying molecule, but it actually has structural roles as well because it holds the chromosome together. It's the polymer that holds the chromosome together. And you have sequences here, like these long repetitive sequences that have a structural role. So centromeres and telomeres are the parts, telomeres at the ends of the DNA chains that signal this is the end of the chromosome. Centromeres and various other proteins that involve, or various other sequences that involve knotting two strands of the chromosome together because the chromosome has a very hierarchical structure. The DNA is wrapping around histones, and then there's another order of structure, and they finally form a nano structure called a chromosome. All this has historically been referred to as junk DNA. It's DNA that's in your genome because it's very good at replicating itself for one reason or another, although there has been some research recently that shows that some of this formerly referred to as junk DNA actually does have some valuable, some value. Within that 1.5% that's protein coding, you get a lot of different kinds of proteins. So for example, transcription factors that tell you, when and where you should transcribe DNA to form an mRNA, signaling proteins from one cell to another, transport proteins, membrane proteins that allow things into and out of the cell that kind of lie like a straw in between the phospholipid bilayer, extracellular structures as well. Okay, so this is what the single nucleotide polymorphism genotype looks like. Suppose you have 4 individuals that are the same pretty much everywhere, like all human beings are, except for one SNP. This is a single nucleotide polymorphism where there is some difference between individuals. So if you take all the SNPs and you add them together, you get something called a haplotype. And it turns out that haplotypes tend to be inherited together, which makes sense because they're close on the same chromosome, and probability is that they'll be inherited together. This is the first page of 62 million entries for my SNP genotype. You can't really get any information from this, so don't try to blackmail me. And what it told me, so this is a company called 23andMe. Has anybody done 23andMe? A few people. So 23andMe, it used to be a lot cooler than it is now, but back when they founded 23andMe, you would spit in a vial and you would send it to 23andMe, and they would do your SNP genotype using Illumina machines, of course. Because Illumina is actually something like 90% of all genetic data that has ever been produced has been produced on machines made by Illumina, right here in San Diego. So anyway, you would spit in the vial, and there would be enough DNA in there from your inside of your cheek cells and all the other crap in your mouth. And they separate it from the bacteria DNA. They get just the human DNA, and they gave you they sent you the raw text file of your SNP genotype, which is really cool. The day that I did it, they had a 1-day sale where it was free with a 1-year subscription, so I did that. Then they give you updates periodically on what diseases you're likely to be susceptible to. And the FDA didn't like that, so they said that you can't, to 23andMe, you can't do that anymore. So now it's just an ancestry service, which is not nearly as cool, but the government doesn't like to trust you with information about your own body. But anyway, I did it before that rule was in place, so I have it, yeah.
They actually have some independent, third-party systems that you can run all the raw data through that'll tell you the same information now.
Oh, that's awesome.
About that.
So just for you listening at home, you can actually send your SNP genotype to a third-party service, and they'll give you all the same information. So that's cool. And one of the things that I found out about myself is that I'm intolerant to caffeinated coffee, which, believe it or not, I didn't have any caffeinated coffee today, but or basically any day. And then it also told me that I also have the sickle cell trait. And the sickle cell trait is something that is, if you have 2 copies of the sickle cell gene, it means that your hemoglobin has a hydrophobic patch on the end of it that allows it to polymerize with other hemoglobins. And when the hemoglobin molecules polymerize, they turn the blood cells into a rigid shape that looks like a sickle under a microscope. And this happens in particular in hypoxic conditions and dehydrating conditions. So if you are working out in the heat or something and you have sickle cell disease, you will have an attack. People that have only 1 copy of the bad gene or the sickle cell gene are not supposed to have any problems except under circumstances that I'll tell you about. So It happens that most people that have the sickle cell disease have what's called an autosplenectomy by the time they're only a few years old. So what happens is that the sickle cells impact in your spleen, where the blood flow is constricted just based on the fact that that capillary flow in the spleen is restricted, and the spleen basically has several infarcts where the blood can't reach the tissue, and the spleen basically dies. So that's called an autosplenectomy. Why did the sickle cell gene persist in the environment to begin with? Well, if you have just one copy of the sickle cell trait, it makes you resistant to malaria, and you're half as likely to contract malaria and 10 times less likely to die from malaria if you do contract it. So that's why the sickle cell trait is predominant in areas that are now or have historically been afflicted by malaria. So this is me. Darren Lipomi has one copy of the S, that is the sickling version of the hemoglobin gene, because my dad is of Sicilian ancestry, and there was malaria in the Mediterranean region historically. Oh yeah, that's for later. So I learned about this from 23andMe right around the time when there was a playoff game between where the Pittsburgh Steelers were at the Denver Broncos, and it was high altitude, and one of the players, Ryan Taylor, had an altitude-induced splenectomy previously. So his spleen became enlarged and he had to have it removed. And what did he have? He had one bad copy of the sickle cell gene, of the hemoglobin gene that made the sickle cells. So then it turns out, I looked it up, that indeed this has happened. But this is, I learned about this a week after booking a trip to Peru to hike to Machu Picchu, which my wife always wanted to do. So I threw caution to the wind. I'm a scientist, so I looked in the medical literature and found that there were only 42 cases of high altitude induced splenic infarct. Now those are only the ones that are reported. So we went to Peru, and I developed a stabbing pain in my side. And to make a long story short, too late, I had a splenic infarct and hiked the entire Inca Trail without eating or drinking anything for 3 days because that made the pain bad. But it did allow me to go experience the Peruvian medical system, and gives you some ideas about how things can work in other places. Basically, you go in the emergency room, the doctor sees you, he or she orders the tests, then you have the tests, and then you swipe your credit card, literally, and you pay, just like at Starbucks, and then they give you a report, and they told me that I was good enough to get on a plane and fly back and see a hematologist at home, which I did. And he said, what you experienced was so rare that even if I knew you had the sickle cell trait and were going to 14,000 feet, I would not have advised you not to go. So I felt good about that, at least. 23andMe will also give you your Neanderthal ancestry. So because there was interbreeding with this previous form of humans called the Neanderthals that went extinct a few thousand years ago, possibly because of modern humans. I'm 2.5% Neanderthal, which sounds like a lot, but the average user is 2.7%. For better or worse, I don't know. And then you can get the map of human migration from what is believed to be the origin of anatomically modern humans in modern-day Tanzania. And then it tells you where the people where your DNA is most similar to where the people are from now. And because my father, like I said, is 100% Sicilian, so it gives me 43% Italian. Okay, that makes sense. And then the rest is non-specific Southern European, which is partially my mother, who's half French and half Irish. And 100% Darren Lipomi. Okay, I think we're out of time, so we may or may not do this next class, but I want to talk about nanoparticle-based drug delivery and possibly some tissue engineering for the rest of the week to finish up our bio-nano introduction. Thank you.